Usually I talk about medical conditions, but today I think I will show you, present you new stuff. I think a lot of it is stuff you've not seen before. And this comes up from our research work within our organization and with collaborators. Now, what are we trying to achieve here? The goal is to get the best brain you can have. It doesn't matter whether you have a condition or you're healthy, and you'll find something fascinating. Now, PBM in general, you've been through sessions and it does work and generally is safe and helps to various degrees for various people. But what we want to do is see if we can get the best possible outcome. And it could mean doing certain things with the parameters. Right. Now, for the biomodulation, we get a bunch of evidence at different layers over the years. It started with cell culture, and you end up with like, okay, maybe 3-4 joules, the best, and so on. Then it went on to animal study, handheld. But I think today, we are ready for hands-free. And we are looking at directing to the brain through transcranial and intranasal. Now, I will show that we are moving on to possibly finding a way to control the various parameters that actually work through a very convenient interface by using artificial intelligence. It's a buzzword today, but we want to see how this is, going to be used in practice. So we have a bunch of things going on, a bunch of research. I have not covered everything here, but you can see, I'm going to draw lessons from what we're learning on how the parameters work on the brain. Now, we have ongoing study for photobiomodulation effect with real-time fMRI, real-time EEG, reading electrical signals from the brain, real-time effects on meditation. This is quite novel. It's super interesting, and you really want to know what we learn. Then we have cell culture studies on tubulins, on living cells like cancer cells, and see what the effects are. I'll talk more a bit later. And then we have collaborative clinical studies with University of Utah on traumatic brain injury, Boston VA, traumatic brain injury, St. Michael's Hospital at the University of Toronto on dementia and mild cognitive impairment. And then brain fog study on long COVID. That's hopefully we'll wrap up pretty soon. Now I want to not talk too much about the clinical studies, but on what we're getting with parameters. Now this is something I am, the principal investigator, Hannah, is somewhere here. This is a PhD project, but we're learning really very important things with finding what's happening to the brain in responding to laser, photobiomodulation, to the head and up the nose in real time. We have plenty of slides, a lot of data, more data is coming up. But here is a slide that came out of Baycrest and University of Toronto, made a poster presentation, so I can talk to you about it. But what you see here is something that I've not even noticed in other forms of brain stimulation, like transcranial magnetic or TDCS and a bunch of other things. So photobiomodulation does have an effect. You don't feel anything, but the brain is responding really quickly within 3 minutes. Here you see 3, 4, 5 minutes in real time, and it's global. Now here's where parameters start to come in. What are we noticing when we start breaking down the parameters? So Baycrest tested 1064 nanometers laser, 810 nanometer, and looking at the bowl response, blood oxygen level dependency, and look at the percentage increases. I'll summarize and just say, if you look at the left column, right? This is just on the transcranial on the right temple. The peak is at 150 milliwatts. If you look further to the right of that particular, top left box, when we go to 250 milliwatts, it starts to get negative. When it's too low, below 100 milliwatts, it doesn't seem to do anything. So there's a very clear biphasic response here. When you talk about biphasic response, how do we figure it out? This is like the second subject or first subject. Baker is going to assess 45. So hopefully we'll finish with the data and all that by the end of the year. Then the intranasal is super interesting. I've got reports in early years, I don't think intranasal is going to work and so on. But just imagine the nose, there's nothing much that's blocking the photons from the intranasal applicator right to the brain, right? Now, there's actually higher percentage response from 9 milliwatts, higher than 150 milliwatts on the temple. So it's about 15 to 20 times more efficient going up to the nose. So just take that with you. And it's quite specific, 9 milliwatts seems to be higher than 5 and so on. If you do it in voxel counts, you come to about the same kind of findings. So just summarize this. From what we're seeing, very quickly, the response from the brain is quick and global. There's significant effect from parameters, and it's quite specific to wavelength. Power density, which is, you can talk about surface irradiation landing on the surface. Pulse frequency, we are testing 10 hertz, 40 hertz, but I'll talk about some super interesting things later on other pulse frequency. And intranasal independently has powerful effects. Now, what are we learning from EEG? We published this in 2019, and it's highly cited because it's quite novel. We deliver gamma at 40 hertz, 40 times a second to the brain, and see what happens pre and post. Now, what we see is the green line is like, you can take it as the baseline, and green is usually, in EEG, QEEG is normal. But red and yellow is you get more stimulation effect, and blue is downregulation. So by delivering 40 hertz, we upregulate 10 alpha, beta, gamma, faster frequency, and downregulate the slow of waves. It has implications for like ADHD where you have the opposite characteristics. So that's quite a bit of implications there. Now we haven't stopped. We are still experimenting, and we are doing real-time EEG. That was pre and post. Now we're doing real-time. We're it's ongoing, but I'll take an example here. This is quite typical in how the gamma is effect. And I'll summarize by just saying that when we deliver gamma 40 hertz to the brain actually does elevate gamma. So if you want to elevate gamma because you heard it improve your lymphatic system flow, or with say cognitive impairments, because of a lot of publications coming up. So that is one way of doing it. Deliver gamma, and you get consistent upregulation of gamma. So these are lines that compare with baseline at the top. The second row is like you put on the device and you're not turning on. The third row is turning on. And you see the third row, the elevation of gamma on the right column there, the last column, you see elevation of gamma. And then we stopped, 5 minutes, everything downregulated. But after we stopped, 15 minutes later, they went up again. We do measure half an hour later, and they still keep going up. So that is one thing. And I thought that, and I've been telling people, this is EEG is really a byproduct, But this paper just came out a few months ago from MIT. He said, no, the brain rhythms are not just byproducts. They are critical in organizing your brain, which means that if you can influence the brainwave patterns, you can actually reorganize the brain. So just, you're saying gamma rhythms on the superficial layer at the top of your neocortex. What you're doing, stimulating one, you're influencing your neighboring neurons. And it's a bottom-up effect in those layers. So summarizing, brain oscillation can be selectively modified by pulsing photobiomodulation to the brain. Response is quick, and brain rhythms have specific characteristics, which opens the door for what we might be able to do to modify the brain. Now, we do cell cultures. When I went into, okay, let's go test tubulins, microtubules, the form structures of your neurons, the axon particularly, see what happens. And we were also testing on living cells. Basically, living cells are cancer cells. We were testing on HeLa cells, which is quite, you see that in the literature, and glioblastoma, the brain cancer cells. And they're relevant. So what do we see? Now we published this. I don't know whether many people in our field take notice, because this words about tubulin, microtubule and stuff can be sounded over the head. Actually, it's not. They're very important findings here. We pulse at 10 hertz, 810 nanometers in vitro. On the left, you see living cells. What we find was actually increase the resistance of the cells. So kind of living cells in mitochondria can actually build resilience to exposure. The right is interesting. On that part, the left picture is the optical density, pretty dense, using a microscopy. On the right, when we pulse at 10 hertz and 810, it starts to separate, which means that the tubulins are disaggregating, separating. And what we see is, and you can see the optical density on the vertical axis is controlled on the top. You can see that the optical density has come down, so it's kind of separated. So it is causing an effect. And this has implications for, we said, okay, Alzheimer's because of beta amyloid, accumulation tau, and then Parkinson's alpha-synuclein. And then there are, some others where you see accumulation of byproducts in the brain. Then, now what does this mean when you go to try to do a gamma? On the left there's a publication that says that actually gamma frequency activates inhibitory neurons, probably GABA. And this is not published yet, but we see with the red ball, when we do gamma, it's different from alpha. It seems to be migrating, switching over. And the bottom right is when we go to 120 is more, 1000 hertz, it looks different. It looks opposite from 10 hertz. So there is the repolymerization again, reaggregation of the tubulins to reform microtubules, and that helps to support the axons, the neurons. And then we published this, I think quite recently, June. Raman spectroscopy is a method to look at the molecular protein structures, and we actually see a shift in the substructure of the microtubules from alpha helix to beta sheets. So there's a transition going on. It's like, alpha is opening up for other treatments, if you like. So summary, 10 hertz is causing transitioning in the microtubule, 40 hertz is consolidating and starting to polymerize. The implications is just a one-liner there. Pulse frequency influences outcomes. You go down to the neurons. Now what about healthy brains? So this is a change, so we're not really talking all medical here, but maybe you want to improve your IQ, function better. If you're an athlete, get ready for a performance in the field, whatever. What are we able to do? Many factors prevent optimum brain performance. The this list is pretty long, so there are many things, everything from traumatic brain injury to diseases, psychiatric complications, depression, PTSD, certain insults even in your body like heart attack, environmental factors like your food, diet, sleep, everything, and your, the physical limitations like the brain size, cognitive factors when you're thinking negatively, then it has an impact. Social and psychological factors like your education, what you're exposed to it when you're growing up. A genetic, we thought about genetic very briefly in other sessions. Actually, it does have an influence. Otherwise, I can say, okay, I can do this, and you become like a superhuman or something like that. We have a limit. That is your set point we're born with. Maybe you can do some interesting things, as I will show you in meditation, but it's, your genes influence 50 to 70%. Of your cognitive abilities. Of course, aging has an impact. So I say generally, photobiomodulation helps with these factors. For me, that's taking care of basics, because we do find that when you do photobiomodulation, like traumatic brain injury, it really does help. Let's look at head towards some extraordinary things. Now, talking about intelligence-related structures, these are all relevant, stimulating mitochondrial activity. As you probably know, repeating what's been said before, growth factors like BDNF and IGF, overall energy, metabolism. Many of you are already experts in this, reducing inflammation, oxidative stress, and enhancing cerebral blood flow, delivering oxygen to your brain. Now, touching on genes, actually a lot of your genes are non-coding. About a very small percent, 1.4%, does this protein coding that leads to changing your level of cognitive ability or intelligence. So this is just saying if you're impaired or you're not, you're born a certain way. Generally, the characteristics of thinner cortex, shorter dendrites, but those who are performing better in cognition have thicker cortex, larger dendrites. Then you're able to transport more information and more quickly. So genes have a role to play. Yeah, I just want to say that. There is a paper being written now for traumatic brain injury patients. I can't show this because it's being written. We can actually see the changes through DTI, diffusion tensor imaging, the regrowth of the axons. So we're talking about regrowth of white matters. So just watch for the papers. It's coming out of the University of Utah. Yeah, this is a repeat, so I won't have to go through and just say that the dendrites actually have a big influence on the performance of your brain. Now, I've been talking about unlocking. What about unlocking the brain? I just take 3 examples of how photobiomodulation has an effect, right, of unlocking, try to fulfill the brain potential. Creativity, now this is interesting, take note. When we are focusing on the default mode network, we are not necessarily always stimulating the default mode network. Actually, as I will show later, if you inhibit the default mode network and move on to the other network, you will kind of release the brain to perform better, right? The default mode network are certain nodes of the brain. I will show that later too. So this paper was published in 2023, and it shows that the creative thinking is better after photobiomodulation pointing to the default mode network. So I just said, am I contradicting myself on the default mode network? And here, this work seems to be having an effect. But actually, they use the gamma, pulsing at 40 hertz. So actually, it is kind of inhibiting the default mode network. And these are measures of creativity. Generally, the measures of creativity has gone up by doing this method. So that is one study to point to that. Meditation, as I've said, is really interesting. We have two studies going on simultaneously. One is in collaboration with University of Arizona. One is in Toronto. And a little bit different. We both use EEG and look at these like psychedelic, looking based on psychedelic measures and see how the brain is performing. So now generally, those in meditation and advocating for meditation, there's enough literature to say that it helps to improve the connectivity in your brain, like hippocampus to parts of the default mode network, right? Then this is often, I think maybe because the illustration is kind of, give a summary, but I'll just summarize that here, looking at different kind of meditation, particularly on, say, focused attention, but is suggesting that the default mode network is deactivated. Then there are claims that meditating on compassion, like the Buddhist method, loving kindness, actually elevate the gamma level by 700 to 800%. So this fast frequency has great relevance on the effect of meditation. And here's another one. Looking at Himalayan yoga tradition, meditation, Isha Shunya yoga meditation, Vipassana, which is quite popular. And as and the versus control, the upper line is actually control. Sorry, the lower line is actually control. As the frequency goes up, all this meditation, generally they follow a pattern that the effect is higher than control as the frequency gets higher. So a summary from meditation literature. There are many styles of meditation, each with different emphasis. There's the alpha, theta, alpha, gamma. Theta is a slow wave. Actually, they seem to be linked. This particular slow wave between the 4 to 8 hertz seem to somehow link to the higher frequency like gamma to help in meditation. And generally, very experienced meditators have high level of gamma. Beyond 100 hertz, you actually cannot measure with EEG, but the trends are there. So high frequency has superpower, but it's individualized. And there is a sweet spot somewhere for each meditator. Now, what is it? What is the button? Now, we were analyzing our data. What are the novel's findings? So we have a device called a NeuroPro, which can pulse from 0 or 1 to 10,000 hertz. So we are able to do a sweep, different frequencies. And what do we see? We're really exploring the pulsing effect on the meditator's brain. So this is preliminary. We're still analyzing the data. But the pattern is like all over the place. You look at the second box, the middle box. Now, where do we see the significance? But if you look at, these are all the subjective self-reporting from the advanced meditators. Now, there are 5 conditions here, from slow up to fast, and number 5 is sham. So this is interesting. You look a bit busy. Let's look at an example of subject 1, the top 2, and subject 2. And it's a bit small, but we look at the data during and then post as a ratio to before meditating. Now the top right box, this guy, the purple vertical line is actually condition 1, which is theta. Now the last row is gamma. So in this particular person, when we deliver theta, his gamma went up. Isn't that strange? But subject 2 is so different. If you look at the purple, in his case, that is actually the gamma. When we deliver gamma, all his is right across the board generally, delta, theta, alpha, gamma, and so on, all went up. So it is individualized from person to person. So we have a lot of work to do to try to, try to see what actually increases the outcomes. Now, what we found is, very interestingly, anyway, this is in the literature, in meditation, in imaging, the posterior cingulate cortex, which is part of the default mode network, is inhibited. Isn't that interesting? Then we did something called dipole analysis to look at the source of where these signals are coming from. And we found that inhibition, this is a technique in EEG actually, advanced EEG, is also coming from the posterior cingulate cortex. So our findings in the literature are aligned. And we can, with all this coming together, 40 Hz seems to be inhibitory, which is not a bad thing. And when you target a default mode network, you're probably going to start to deactivate it. And what's happening here? So the literature in meditation says that when a person is in higher state of meditation, his default mode network is actually deactivated, and he starts moving into the salience network, the central executive network, and the central executive network and default mode network are anti-correlated. They don't like each other, but the salience is like a bridge. So that's why you shut down the default mode and move into the central executive. That means if you're able to affect locations of the default mode network, then we might get this effect of elevating brain function for meditators. So they report a meditation bliss at a certain trigger, at a certain frequency that takes them there. And we have different, some people at 40 Hz to get there, a lot of people at 120 Hz. The advanced one seems to be 200 Hz, and the investigator in Arizona, he's a really advanced meditator. For him, it's 1,000 Hz. He's been trained to, he started like from 200, he went out to 1,000. So pulse frequency really doesn't matter. So in summary, we're targeting the default mode network, which is highly relevant in meditation, but you are either stimulating or inhibiting. And for meditation, we are looking to inhibit the default mode network with faster frequency. Okay, that's another summary now. Now we move into something very interesting. When I talk to sports mental performance coaches, everybody says, okay, trying to get into the flow. What do you mean by flow states? Some of you might be familiar. And it is really an area where you have high focus, you are kind of in your zone. Zone is another word being used. And you perform, your brain perform at a high level, right? So what is a path to the flow state? It all started with a professor who's originally from Hungary. I can never pronounce his last name, but Mihaly is easy to remember. So he started this in 1975, right? So, and this is kind of representing what flow is about, right? It is elusive, and this paper suggests that maybe you can do this with brain stimulation, but they were talking about TDCS, electrical stimulation. Someone's got to teach them photobiomodulation. So it needs focus and repeated practice for them to get to the flow state, right? So that it comes more easily. This is quite you know, okay, this is a busy slide, but generally they have the same idea using fMRI, structural MRI, FNIRS look at the blood oxygenation in the brain, EEG, and measuring the cardiovascular, so on. Now, fMRI, you can see the first line, flow experience associated with lowered activation of brain waves associated with the default mode network. So even in flow, you want to deactivate default mode network and move on to another state. How do you do it? I've already showed you. You do it at a higher frequency, gamma and above, right? Now, this is a recent paper that has got quite a bit of interest in those who are looking for performance among musicians. And they found that the expert musicians, as opposed to beginners, actually kind of go into autopilot and let go. And same is sport, you're thinking too much, you're not going to perform that great. And then this other people say, oh, you got to keep training and get into goal setting and get high level involvement and so on. Practice, practice, practice. But is there an alternative approach, right? Now we're looking at lower alpha from various papers, fast frequencies, higher frequencies stop at the top layer, so you get faster processing. And then we are able to get into a point when we can start developing a protocol for athletes. And we are actually working with elite athletes and proposing this protocol right now. And here's a paper that's kind of almost to say, you're trying to make your brain, your neural networks more efficient, neural efficiency hypothesis, basically better connectivity, less effort, and so on. So it gets back to the same thing, right? Deactivate the default mode, move on to your salience and central executive. So this is putting together a simple protocol where we say, okay, before your event, the performance, but 30 to 60 minutes before, we use our device, the NeuroDuo, the gamma, plus the intranasal, which is red at 655, which is a red laser. And then after the event, when you're resting, recovery, use 10 Hertz because it helps to consolidate what you're learning. And alternate days, alpha and gamma. So now, who are using it? This is spotted in Instagram before we got into communication. This is Ilya Tupura, who became the world featherweight champion in, I think, February, using our device for his training. Then he won. He has another fight, I think, in October. Let's see what happens. So he's one guy who actually figured out the protocol himself. I said, hey, it's the same thing as what I would have recommended. And then Alicia Newman is like celebrating in Canada. For the first time, we won a medal in pole vault, but she had attempted in 2 Olympics. The problem was she had traumatic brain injury. Now she has overcome that, and that actually helped with her performance. Now she won a bronze medal. So PBM in general can get you better. Try to personalize it. And this is a summary. Looking at real surface irradiance. Now we're still doing research. I got to qualify here. And this is what we see so far. The peak seems to be 150 milliwatts in surface irradiation or power density. We're using laser in the transcranial and 9 milliwatts in the nose. Positioning on a default mode network, pulsing at 10 and 40 Hertz. Let's see what we can do for the best. Now, these are within reach, trying to reach, unlock the potential. We recognize that, we can either stimulate or inhibit. There is a pulse frequency specific between individuals. You can select the LED on or off in certain positions. You can strengthen or weaken the coherency between 2 points. And then cross-frequency coupling. This is one area we are actually experimenting and see we can couple theta and gamma together. I don't have time to go too deep into this one, right? And then we have a tool to play around with different parameters. with this device that I mentioned, but we are not there yet. So the future is really in artificial intelligence. We are going to use EEG to guide the closed loop, machine learning and artificial intelligence to personalize the right parameters for individuals. Now I got this published, you see the data put there, it's 23rd August, like yesterday. Okay. Now I want to point to this because it actually follows what I'm trying to tell you, look at ways to, optimize the parameters. And this is a model looking at all the literature. I synthesized into this model. Go to it, it's out there, it's open access. And the stars are pointing to where, based on the literature, all the work done in the past, the photobiomodulation can affect, these various aspects of the pathophysiology of Alzheimer's disease. So we have that potential here. And I'm suggesting that, photobiomodulation can penetrate the brain, but the avenue that we can look at is look at the waveforms of the brain. Like I said, you can modify, the structures below and the networks. So lots of people to thank, lots of collaborators. I'm sure I haven't mentioned everybody, but thank you for attending.